You know, that fundamental story of biology we all learned in school? It turns out that might just be chapter 1. We're taught that our genes are basically the blueprint for life. But what if I told you the real story is way more complex, a lot more creative, and just full of secrets we're only now starting to figure out? Okay, let's just dive right into it with a question for you. We've got around 20,000 genes that code for proteins. Seems simple enough. But if you take just one of those genes, how many different functional proteins do you think it can actually produce? One? Maybe a couple? Go on, take a guess. Well, the answer to that question kind of forces us to bust open one of the most famous ideas in all of biology. You know, a concept that's so foundational, it's pretty much treated as fact. But as we're about to see, it's a really beautiful, elegant, and, well, a profoundly incomplete picture of how things really work. So this is the classic model, right? The one from Beadle and Tatum way back in the 1940s. One gene holds the code for one protein. It's the simple linear factory model of life. DNA makes RNA, and RNA makes a protein. Story's over. And for decades, this was the map we used. But even back then, early experiments were hinting at something more. They were seeing these weird smears and extra spots on gels where really there should have been just one. It turns out those little experimental quirks, they weren't errors at all. Nope. They were clues, and they were pointing to this hidden universe of incredible complexity. So it's time to update our map and step into the much more exciting, much more dynamic reality of what we now call the proteoform universe. Okay, so back to that question I asked, how many different proteins can one single gene make? The answer, on average, is around 50. Yeah, 50. 50 distinct molecular versions from just one single genetic recipe. And this chart, this just brilliantly shows the fallout from that diversity. You take our 20,000 or so genes, and through this incredible process of variation, they literally explode into an estimated 1 million or even more distinct protein variants inside our cells. Just look at the scale here. This isn't some tiny little difference. It's a complete biological revolution. So obviously, we need a name for all these different versions. Scientists Lloyd Smith and Neil Kelleher came up with the term proteoform to describe every single one of these distinct molecular proteins that can come from a single gene. And you know, this isn't just some new vocabulary word. It's a whole new way of thinking about the machinery of life itself. So how does our biology even pull this off? I mean, how does one blueprint create such staggering variety? Well, probably the best way to think about it is like having a master recipe that you can tweak and customize to make a million different meals. And there are 4 main ways the cell does this. First up, you've got genetic variation. This is like changing the core ingredients in the recipe, a mutation in the DNA itself. Second is transcriptomic variation. This is where the cell edits the RNA instructions, a process called alternative splicing. It's like tearing out a few steps or swapping paragraphs in your cookbook. Third, there's translational variation. Here, the cellular machinery actually reads the recipe in a new way, maybe starting on a different word or shifting how it reads everything. And finally, the icing on the cake, post-translational modification. This is the final garnish. After the protein is already made, the cell sticks on these little chemical tags that can completely change what it does. But here, this is where it gets even more fascinating. These 4 layers of diversity, they don't just exist on their own. No, they combine, they interact, and they produce results that are just way more complex than the sum of their parts. So these interactions can be synergistic, where 2 changes actually amplify each other's effects. They can be antagonistic, where one basically cancels the other one out. They can even team up to create a totally new function that wasn't there before. But maybe the most mind-blowing is a restorative effect, where one defect can actually compensate for and fix another one. And this leads us to one of the most amazing stories in proteoform biology I have ever heard. It's the case of the CFTR protein, which is linked to cystic fibrosis. And it's the perfect example of where 2 biological wrongs can literally make a right. Okay, so here's how it works. First, you get this one mutation that causes the CFTR protein to misfold, making it totally useless. That's problem number 1. But then a second, so-called silent mutation shows up nearby. Now, this one doesn't change the protein's building blocks at all, but it does one absolutely crucial thing. It slows down the ribosome, the little machine that builds the protein. And this slowdown, this gives the faulty, misfolding protein just a little bit of extra time to fold itself into the correct shape, completely restoring its function. It's incredible. And the source material just puts it so perfectly. This silent change gives it the extra time it needs for correct folding. It's this beautiful example of what's called intragenic epistasis, where the effect of one genetic variant depends entirely on another one being there, right inside the very same gene. So this all brings us to the ultimate point, the future of medicine. I mean, think about it. If a disease is often caused by one specific faulty proteoform, out of 50 possibilities, how can we possibly hope to treat it by just looking at the gene? The answer is, we can't. The future of really effective, targeted treatments isn't just in our genes, it's in our proteins. We have to move past the blueprint and start focusing on the actual molecular machines that are doing the work. And that means we have to identify and target the exact proteoform that's causing the problem. And to get there, researchers are using this really powerful three-step framework. So first, they map the entire landscape, creating an atlas of all human proteoforms with amazing tech like top-down mass spectroscopy. Second, they perturb them. They systematically break or change individual proteoforms just to see what happens. And finally, they use all that data to predict the function of the millions of proteoforms we haven't even tested yet, figuring out which ones are most likely linked to disease. You know, this just opens up a whole new frontier. We've gone from thinking about one protein per gene to this massive universe of over 1 million variants. It really makes you wonder, what cures, what treatments, what fundamental secrets about our own health are just hiding there in plain sight, waiting to be discovered in our proteome? Thanks for watching. Please like and subscribe Computational Genome Biology.